We're going to talk about DNA and RNA, as well as the process of duplicating, copying that, and rewriting DNA into RNA, which is transcription, and then taking the RNA, bonding amino acids together to make proteins at the ribosome, and that is translation. So let's take a look. The first thing I want you to know about DNA is a short word for deoxyribonucleic acid. Deoxy, if I put the prefix D in front of something, it means taking away or having one less. So we have one less oxygen in the ribonucleic acid. So DNA has less oxygen than RNA. So that's why it's called deoxyribonucleic acid. It has one less oxygen than the RNA does. So DNA is a double-chained, just like a ladder, has two sides of the ladder, a left and right side. DNA is double-sided. It's a nucleic acid. Remember, we talked about proteins, carbohydrates, and lipids. Now we're dealing with nucleic acids. Now, nucleic acids are going to be containing these bases, and we'll talk about those four bases in just a little bit. But DNA is a double-chained nucleic acid that is made or that is located in the nucleus. Now, the process that we're going to be learning about transcription and translation comes out of the nucleus to do that process, and we'll talk about that later. But DNA contains all our genetic information that gives us all our features and produces all the protein structures of our body. So this is very important. So you can see how DNA is wrapped up, it's twisted, it has your two sides, okay? That's your double helix. Now here's a better picture. This is a more accurate picture. You can see the left side has a phosphate, that's the P, then it has a pentose sugar, remember a five-sided sugar, and then you go down that ladder, we go phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And so that's one side. Now, if you look at the other side of the ladder, the other structure, we still have phosphate sugar, but look, the phosphate and sugar, that sugar is upside down. So in DNA, it's pretty interesting that one left side and right side, they're flipped. They're kind of opposite each other. Now, right in the middle where we put our feet on the ladder, those are our base pairs, or nucleic bases. So we're going to be talking about the four nucleic bases, adenine, thymine, they go together, A and T, and then guanine and cytosine, G and C, they go together. So here are the three components. The phosphate, okay, made up of the element phosphorus. Then we have a pentose, a five-carbon sugar, a five-sided sugar. And then our nucleic base pairs. A with T, G with C. Now let's look at RNA. RNA is ribonucleic acid. Remember, DNA, deoxy, had less oxygen than ribonucleic acid. So what is RNA? RNA is a nucleic acid just like DNA, but it actually copies the information from the DNA and makes proteins. There are three types of RNA we're going to talk about. RNA has base pairs just like DNA, but the problem is one of the base pairs are different. So RNA does have adenine just like DNA does. It does have cytosine. It does have guanine, but RNA does not have thymine. RNA has uracil instead of thymine. So when it goes to bond or copy, okay, in forming RNA, it will not have a thymine. It will have a uracil. And we'll do some practice with that later on to help you understand that more. The first type of RNA is called mRNA, or messenger RNA. This looks like a little train. This little train is not like DNA, where DNA is double-sided. This is single-sided, only has one part, only has one side. It's a little train. It will actually copy, it will cause DNA to unzip. Okay, it will copy one side of the DNA, and then it will travel to the ribosomes to make the proteins. The second type is the ribosome, the rRNA, or known as the ribosomal RNA. So ribosomes are actually made up of RNA. And this actually is kind of like the train station. This is where the mRNA, the single train that copied from the DNA, comes in. It will come into the RNA, just like a train comes into the train station and waits for the people to come in. And that will be the tRNA. The tRNA is short for transfer RNA. The transfer RNA brings in amino acids to the ribosome. They hook in to the mRNA. So the tRNA hooks in at the mRNA. So the tRNA is going to come in and bond and attach to the mRNA, which is located at the rRNA. So the tRNA are like the people getting on to the train station, okay, getting on and hooking into the train at the train station. So the tRNA, the people, hooks to the train. They go into the train, okay, at the rRNA, the train station. Okay, here's a picture of the mRNA. You can see it's single strand. The rRNA, that little blob, that ribosome, and then the tRNA. And at the very bottom of the tRNA, you can see it says anticodon. And we're going to talk about this in class, that three little parts of the mRNA. If we just look at mRNA at the top left, it says A, C, G. A set of three would be a codon. Well, that codon needs to match up with the anticodon of the tRNA. So they are going to bond together. We'll practice this in class. So let's talk about this. Here's the first step, transcription. This is when the DNA unzips, so we have that double helix, it unzips, and the mRNA is formed, is made by copying the parts of the DNA, only one side of it. That is called transcription. Think about the word transcription is rewriting. If someone transcribes something, they rewrite it. They rewrite it. They kind of put it in their own words. Okay, so only one side of the DNA is being copied. Now let's look at the picture. Let's look at the top left of the DNA. Do you see the red G? Red G? Well, the mRNA is going to have the same nucleic bases, remember, except for thymine. It'll have uracil. So G goes with C. Okay, that's correct. Now let's go below that red G, and you can see there's a blue A. Well, on the mRNA, remember, mRNA does not have thymine. It has uracil. So instead of thymine, we're going to put a uracil. So as you look down there, G goes with C, C goes with G, but A will always bond and make a U. Now if we go down and we see the T on the DNA, on the mRNA it will make an A. But on the DNA, the A will make a U of messenger RNA. And if that's a little confusing, we will practice that. We'll do some labs with that. This is something you'll learn in high school about RNA polymerase. Polymerase, if it ends with ASE, that's an enzyme. So basically, this is going to start the transcription process. This is going to say, okay, we're going to start doing this process. So it actually starts the unzipping. The termination will actually, there will be a stop signal. This will be a set of your codons that will stop the polymerase, stop the transcription process. Okay? So there you can see the word is a codon is a set of three, and we'll be using that a lot in our labs. Okay, a set of three nucleic bases. So if I say ATA, that is a codon. Okay, that's a set of three. Okay, let's look at translation now. So the mRNA in transcription, it put it in its own words. It went from DNA to RNA, and it did that in the nucleus. It now escapes the nucleus, and then it travels through the cytoplasm and finds the ribosome, the rRNA. And as it finds the ribosome, it kind of comes in. Remember, it's like that train coming into the train station. And so you can look at that picture there, and you can see that the tRNA is coming in like little people getting on the train. It's bonding. Remember, we're not dealing with DNA anymore. We're dealing with both of these will be RNA. So the ribosome, the mRNA, and the tRNA will all have uracil. So in this process of translation, it will never have a thymine, never have a T. Now, translation, think about this. We're not just putting it in our own words, we're putting this and making a new language. We're translating it. We're going from a nucleic acid now to protein. So we're going from DNA and RNA, now making proteins. Now, look over that, look at the pictures. If it's not all crystal clear right now, remember, don't flip out, calm down. We're going to do lots of labs, and we're going to do a lot of practice. We're going to do lots of discussion so that you know what's happening in these processes. Okay? You'll do well.